So welcome to the second microbiology spot. This is purely bacteriology. So we start. Question 26. Identify the above presentation and the bacteria that causes it. Name the major toxin responsible for the tissue destruction and its mechanism of action. 2 ways of treating it. And then what culture media can be used in the above bacteria. So I want us to be interactive. Type your answers in the chat as we go through. Any point that you guys mention, I'm going to be explaining, because this session is supposed to cover MCQs, SAQs, and even spot. Okay, so let's have answers in the chat. This is gas gangrene. Yes, you can see it appears that there is gas in between the skin. Okay, underneath the skin. Okay. Caused by Clostridium perfringens. Very good. Then the major toxin responsible for the gas gangrene. Alpha toxin. Very good. Now, mechanism of action. This is very relevant. You can be asked how the toxin works. Okay. It degrades lenticinase. So lenticinase is the phospholipase causing myonecrosis. Yes, very good. Okay. I'm seeing a question. How do you differentiate gas gangrene and necrotizing fasciitis? So for gas gangrene, as you can see in this picture, you'll note that it seems as if there is air in between the skin, the subcutaneous tissue. Okay. For necrotizing fasciitis, how it occurs is secondary to strep pyogenes or Staphylococcus aureus. It literally destroys the skin. So there'll be necrosis of the skin. Okay. I don't know if I have a picture. I can show you guys for necrotizing fasciitis. Let me show you a picture on necrotizing fasciitis. So, okay. Now, you can see in necrotizing fasciitis, let's look at this picture. What is happening is the skin is being destroyed. Okay, so how it starts as a cellulitis, then the skin peels off, then it continues to destroy. So you remember the layers of the skin, we had the epidermis, the dermis, hypodermis. So that's how the skin is going to be destroyed. Okay. That is how you expect the skin to be destroyed. Now that's how you differentiate. But if you see this other one, let me share. If you see for this, you can see that there is some gas. Okay. Can you see it? Hope you can tell the difference. Now, we were saying ways of treating. So I saw some answers, surgical debridement. This is a surgical case. And we have to debride. You take them to theater, you remove these layers, and then we use antibiotics. Then what culture media can be used for this Clostridium? High dose IV penicillin, yes. Hyperbaric oxygen therapy for gas gangrene, yes, that can also be a way. Okay. So here are the answers. Gas gangrene, Clostridium perfringens, alpha toxin, lenticinase, hydrolyzes phospholipids in cell membranes, leading to cell lysis and tissue necrosis. Surgical debridement, broad spectrum antibiotics. And then we have Robertson cooked medium or blood agar. We're going to see double zone hemolysis. So I'm sure there's a question for testing this concept. Then we have thioglycolate medium. Now, I have a question for you guys. So apart from the gas gangrene, what else does Clostridium perfringens cause? Food poisoning. Very good. Late onset food poisoning. So if you can remember, for Clostridium perfringens, it has spores. Okay. Now these spores, if it's heavily contaminated food with spores, and you cook it and you leave it standing for a while, the spores are going to germinate, leading to the formation of vegetative bacteria, which now leads to the production of a heat labile enterotoxin. Okay, so that's why we call it late onset. Usually occurs around 10 to 12 hours. Okay, the food poisoning symptoms. Food poisoning symptoms are like diarrhea, sorry for that, diarrhea, vomiting. Okay, that whole aspect of it. Okay, so I hope that's clear. Then we go to this. So I want you to look at these teeth. Okay, can you see these teeth? Can you see these teeth? What do we call this presentation? They are notched teeth. Hutchinson's teeth, very good. Then what bacteria causes this? Treponema pallidum, very good. Mode of transmission to the child? Transplacentally, so via mother to child. And then list two screening tests. So this is a very, very high yield point that I want you guys to understand. Okay, we are going to go through it. So let's see if you're okay. Hutchinson's, if you say notched teeth, pegged shaped teeth caused by Treponema pallidum, they are all correct. Transplacentally or vertical from mother to child. And then they're asked list two screening tests to be performed in this child. Now, before we even mention the screening test, I want us to go through congenital syphilis because it can be asked in very different ways. You can see this patient, they have the notched teeth. You can be given a patient with, we call it Shaba shins. Basically, the shins have a very different appearance. Okay, the Saba shins, sorry, say that 5 times. Saba shins, okay? So what are the aspects of congenital syphilis? Because you can be asked. What are the features of congenital syphilis? There are quite a few. On the face, we have what do we have? Mulberry molars, very good. A saddle nose. These Hutchinson's teeth, okay? A short maxilla, very good. And then what cranial nerve is mainly affected in congenital syphilis? In case I would have asked that, what cranial nerve is mainly affected? Ragades, yes, Ragades is there. Ragades is See these things, we need to see pictures because it's a spot and they can bring for you. So hope you can see my screen. It's a feature of congenital syphilis. Now, can you see the angle of this mouth? Here's that picture. Okay, just observe from here. So Ragades, the angle of this mouth, okay, there are linear scars here. Okay, that is what we refer to as Ragades. Okay, so if you see such a picture on a spot, this is congenital syphilis. And then the mulberry molars, let me show you. Okay, can you see them? They look like mulberry. Can you see them? They are overly notched here. Okay. Yes. Is there another one? Oh, the subachines. I want to show you the subachines, how they look like. So all these features you can be brought for in a spot. Remember, they're all for syphilis. Now you can see subachines. They look like this sword. The sword is called the shaba here. Okay. That is the subachines. Okay. Vestibulocochlear nerve, very good. Cranial nerve, 8. Good. Now, we have a lot of questions, and I want us to cover a lot, but at least you've understood that. Now, let's go to the tests, which are very, very important for you to know. Okay. List 2 screening tests. So in syphilis, we have 2 types of serological tests: the non-specific tests and the specific tests. So here's a good picture. Okay. You can be told to discuss, to list them. Okay. Now, I want you to understand this. These are serological tests. This means that you take a blood sample, send it to the lab. For these ones, the non-treponemal tests, these are non-specific to syphilis. So we have VDRL, CAN test, and rapid plasma reagin. Okay. So why they are non-specific to syphilis? If they are positive, it can indicate you have syphilis. But on the other hand, they can also cause false positive results. So in conditions such as pregnancy, viral infections such as hepatitis, use of certain drugs, for example, procainamide, lupus, all those conditions will give you a false positive VDRL, okay? Then for treponemal tests, these are now the specific tests. If they're positive, you definitely have syphilis, okay? Now, they include treponema pallidum hemagglutinin assay, TPHA. We have fluorescent treponemal antibody absorption assay, FTA-ABS, IgM FTA-ABS used for diagnosis of congenital syphilis. We have ELISA, and then we have treponema pallidum immobilization test. So please know this because it can come as an SAQ where they tell you mention or discuss the treponemal serological test or just in an essay. Okay, so something to note, these tests, the treponemal tests, they remain positive regardless of the disease or even treatment. Okay, so if you have an active syphilis infection, it will be positive. And then if you have the antibodies against the syphilis, it will also be positive. Okay, now I hope that's clear. So please, you have to know these. Then I used here the word screening. So you see these tests, they are treponemal tests and they're very specific. We don't use them for screening. For screening, we do the non-treponemal tests. But for a definitive diagnosis, we use treponemal tests. Okay. Now, don't confuse this with the direct testing. Okay. You see how we do dark field microscopy. We see the spirochetes, which are white in color in a black background. That is direct testing. Okay. That is not serological testing. Okay. Number 28. Yes, please share, answer each other on the chat. Okay. Yeah, those are the pictures you have to know for syphilis. Then, as you can see here, this is a rusty nail in soil. Then you're told, name the organism whose reservoir is shown. Name the exotoxin produced by the bacteria and its main effect in the human body. Describe the characteristic appearance of the organism under a microscope. Okay. So I'm sure you've all known this, that Clostridium tetani, the reservoir is in soil. Okay. And then the exotoxin produced by Clostridium tetani. So as you can see, the Clostridia are very high yield. And we have, is it 4 Clostridia? Tetani, botulinum, perfringens, and difficile. So these are the must knows. Tetanospasmin, very good. So tetanospasmin, how does it work? What is its main effect? Okay. So we have tetanospasmin. It blocks inhibitory neurotransmitters, which are like GABA, leading to muscle spasm. So for a person with Clostridium tetani, they'll have rigid paralysis. We also call it spastic paralysis. Okay. That's why we have a very common symptom, which is lockjaw. Literally, their masseter gets locked. Okay. That is the lockjaw. Okay. So because here GABA has been inhibited, has been blocked, they are unable to relax their muscles. Okay. Then describe the characteristic appearance. So we have the drumstick appearance. This was direct. The gram-positive bacilli. Then remember, Clostridium are spore-forming. It binds to the snare proteins causing muscle spasm. Very good. Now that is the exact pathophysiology. Drumstick, you can also say tennis racket appearance. Very good. Okay. Yeah, I don't think I have anything else to mention for tetany. What you have to know is soil is its reservoir. It produces tetanospasmin, which blocks inhibitory neurotransmitters like GABA, and then it causes spastic paralysis. That's the most important thing to do. And then you do the drumstick appearance. Another issue is we treat. So how do we treat tetanus? How do you treat tetanus? How do you treat tetanus? DPT immunization. So immunization. Okay. Yes. So what happens is, remember, this is a bacteria, so we have to give antibiotics. Anytime you have a bacteria in mind, remember antibiotics, okay? Mostly metronidazole, very good. Now, we have the antibiotics. Anytime you're asked about treatment, remember that. Then now, specific to tetanus, we have to give the antitoxin, which is normally denatured Clostridium tetani toxoid, okay? Then you give the vaccine booster. Okay, then depending on the wound, let's say this person, got very injured, we can do wound debridement, where we remove the dead necrosed tissue. Is it possible to type your question on the chat so that we can move faster? Or you have something burning? Okay, I'm skimming through the chat. Okay, so I hope we're clear on tetanus. Please repeat the treatment. So, number one, if you ask treatment for any bacteria, you can get specific with the antibiotics, but I think that is cooking yourself, because there are some antibiotics, for example, TB. We cannot use penicillin because it does not have a peptidoglycan layer. It doesn't make sense. So you can just mention antibiotics. Now it depends on the lecturers. I was told that some are lenient, I think because of today's meeting, okay? Some are not, but if you mention that, you deserve a mark. And then you now start thinking about, you start thinking of the specific bacteria. So in this case, we have tetanus. So it presents with muscle spasticity. The muscle becomes rigid. So we have to give an antitoxin for the tetanus and a vaccine booster. Okay. Now, the next thing is wound debridement in case you have an injury, because how did the tetanus enter? Maybe you stepped on this rusty nail, then it got into your skin. Okay. If its reservoir is soil, why do we talk about rust? Yeah, that's usually a common misconception on that tetanus exists in the rust. It's not really the rust, it's the soil, not the rust, but it's associated together. Okay. Yeah. Are we clear? And then I'm seeing a question, does tetanus bind to or cleave snare proteins? So the mechanism of action is similar for tetanospasmin and botulinum toxin, Clostridium botulinum. So what it happens, it actually cleaves the snare protein. Okay. I had mentioned bind. Sorry, I hadn't seen that. It cleaves it. Okay. Let's go to this. This is also a very, very high yield spot question. Anywhere you go, you must be asked this. And these are the questions where they are the first questions of microbiology, the first topics that you learned. So let's label this together. What is A? This one's everyone should be singing it. A, B, C, D. What is A? Lag. So it goes lag. After lag, we go to. So we go lag, then. Sorry, I've showed you the answers. Lag, log, yes, exponential. And then finally, death. Okay. So this is how it goes. Lag, log, stationary, death. That's how it is. Okay. Now, the next thing you'll be asked is how the characteristics of each stage. So here they were saying, identify the stage where the following occurs. Fluid gets into cell causing lysis. If you see lysis means cell death. So definitely it's D. Okay. Then you'll be told to identify the characteristics of each stage. You can be told stage A, stage B, or C. Now in stage A, which is the lag phase, the bacteria is adapting to new environment. Okay. So depending on where it is, then here it hasn't started to divide. Okay. Dividing starts taking place here. So no cell division here. Here there is vigorous metabolic activity. They increase in size, but do not divide because they are preparing to divide. Then in the log phase, so how to remember, lag comes before log, A comes before O. So this is definitely lag, then log. So here the cells start dividing and the numbers increase exponentially. It shoots. And there's increased rate of metabolism. That's why the numbers shoot. Then C is where now the stationary phase, the death rate equals the growth rate, which makes sense because here the growth rate is more than the death rate. Okay. Cell division stops due to depletion of nutrients and accumulation of toxic products. Here is where now spore-forming bacteria, the spore is formed in spore-forming bacteria. Okay. And then stage D, just to mention, this is where we have the death. So death is going to occur. Here, the death rate is more than the growth rate. Okay. Cell lysis is going to occur. Fluid enters the cells, leading to a bursting of the cells. Okay. Very clear. Now, we go to generation time. So I was glad when people are doing this question. Can stationary also be called plateau? Yes, I mean, it looks like a plateau. It's a straight phase. So here, generation time, I was proud that people got this. The time required for a bacteria to give rise to 2 daughter cells under optimum condition. Okay. So E. coli, if you are to culture it under optimum condition, it will multiply in 20 minutes. TB will multiply in 20 hours. M. leprae will multiply in 20 days. You see, it's very long for Microbacterium leprae. Okay, so this one, please get to know that. Those are the only things I can test from this diagram. Then we go to this. Now this, I'll have to take some extra time to explain because it can come as an essay. So let's go through it together before I explain it. Technique shown, definitely it's gram stain. Then name the procedures A to E. Okay, so people name, and then what's the function of procedure C and D. So let's name the procedures first before I explain, because it's very, very important. It did come as an essay. Okay, so number A, fixation, good. Then B, crystal violet, good. C, I think you guys did this in the lab, and the lab tech, I remember he was very meticulous. You have to know the exact, he even gave you the timings. 1 minute, 1 minute, 10 to 20 seconds, 1 minute. Iodine, so that's C. Yes, C is iodine. Then D, what is D? Decolorization, yes. And then E is now the last stain where I use there. What do you use for E? Counter stain, very good. Now, let me explain. And please try as much as possible to understand, because they'll ask you. So we start. In a gram stain, we have to prepare the smear. When we prepare the smear, what is going to happen is you take it, place it on a clean glass slide, and then you allow it to air dry. After drying, you have to fix it. So there are 2 types of fixations. We have heat fixation and we have chemical fixation. So heat fixation is where you pass through it, you pass through a flame. Then chemical fixation is where you put methanol. So you can be asked why we do that. Number 1, we have to kill the bacteria. Number 2, sorry, let me take this. Sorry for that. Now, as I was saying, why you can be asked why fixation occurs. You have to say, number 1, we kill the bacteria. Number 2, the bacteria adheres firmly to the slide. And then number 3, through that fixation, we are preserving the morphology of the bacteria, the cell morphology. Now, after fixation, we go to B. Now, here's where we use the crystal violet, which is the primary stain. It's purple in color. That's why you can see, I don't know, is this purple or blue? They say men are colorblind. I'm seeing purple. So here what happens is the smear is flooded with the crystal violet for around 30 to 60 seconds. Okay. So why we're doing this is because we want this crystal violet to penetrate the bacterial cell wall. Okay. And then it will stain it this color, the purplish color. So here, regardless of whether it's gram positive or gram negative, it still appears purple. Okay. That's the primary stain. Then we go to C. Now C is here where we use the mordant stain. So mordant is the iodine solution. So you're done with the crystal violet, you flood it with iodine. After flooding it with iodine for another 1 minute, what is happening here is there'll be a complex of that crystal violet and iodine stain for it to be trapped within the bacterial cell wall. Okay. Now here, both of them will still remain purple. But now, for the bacteria that are gram-positive, which have a thick peptidoglycan layer, is where you expect it to remain purple after the next stage. Okay, but both of them remain purple at this stage. Then we go to D, where we decolorize it. Okay. So decolorize it, we use with alcohol. So alcohol, it can be anything, acetone, ethanol, all those types of alcohol. Okay. Here we just put it for like 10 seconds, and then we rinse immediately with water. So here, now, in gram-positive bacteria, they have a thick peptidoglycan layer. So we expect them to remain purple. But for gram-negative, that alcohol that we flooded the slide with is going to dissolve the stain. And once that happens, because it has a very thin peptidoglycan layer, the dye, which is that complex, is just going to escape. So that's why it's going to appear. Is this colorless? It's supposed to be colorless, not white. Assume it's colorless, okay? And then now the final stage, we now use the counter stain, which is the safranin, which is a pink dye. So you put safranin for another 1 minute, then we rinse with water and we dry it. So the gram-negative bacteria will absorb the safranin and it will remain pink. But for gram-positive, since it was purple initially and it has a thick peptidoglycan layer, yes, it's going to retain that pink color, but what is darker, the pink or the purple? It's the purple. So that purple is the one that is going to remain, okay? So I've explained that. Please remember that. So you can be asked to describe it. You can be asked the function of each state, and I've made it very clear, unless there's a question. So here are the answers: fixation, crystal violet, iodine treatment, decolorization, counter stain. Then here are the functions of the procedures, okay? Now, classify gram-negative bacteria according to shape, giving at least 2 examples. So I saw some answers from you guys. They were not complete. Why I say that is because we have quite a few shapes for gram-negative bacteria. Now, the shape is supposed to help you in terms of knowing the characteristics of the bacteria. Now, there are 4 main shapes. Tell me on the chat. Let me help you guys. We have bacilli. Okay, yeah, more shapes. We have bacilli. We have cocobacilli, very good. We have diplococci. And then we have diplococci, yes, mentioned. Cocci. Diplococci. There's one more. Pleomorphic, yes, but there's one more I'm looking for. Yes, please. It's very good you answer your colleagues at the chat level. Spiral, very good. That's the one I was looking for. Spiral, you can call it spiral, you can call it curved, and all those. Okay. Now, you've known the shapes. Now let's classify. Diplococci, these occur in pairs. Diplococci. There are only 2, and they're both aerobic. We have Neisseria meningitidis and Neisseria gonorrhea. We differentiate them by maltose fermentation. Okay. Neisseria meningitidis will ferment maltose and glucose, but Neisseria gonorrhea ferments only glucose. Okay. Then we go to the cocobacilli. Cocobacilli is now, they are rods and they are curved. Not curved, sorry. They are spherical and rods in between. Okay. That's the shape. So we have Haemophilus, Bordetella, Brucella, Pasturella, all those ones, Francisella. Okay. Then we have the curved. The curved are 3: Campylobacter, Vibrio, and Helicobacter. Okay. And then the last one is now the bacilli. Now here in the bacilli is where we divide them into lactose fermenters. Okay. We have a good way of defining them. We can divide them into lactose fermenters and non-lactose fermenters. So the lactose fermenters are the sick, Citrobacter, E. coli, Enterobacter, Klebsiella. It's actually 6. The mnemonic is 6. Okay, type on the chat. Okay, I hope you're following through, or I'm going too fast. 6: Citrobacter, Enterobacter, E. coli, Klebsiella, and Serratia. And then for the non-lactose fermenters, we can also divide it into two: the oxidase positive ones and the oxidase negative ones. The oxidase positive ones are things like Pseudomonas. Okay, and the oxidase negative are now Shigella, Yersinia, Salmonella. Okay, now that is something you should know at your fingertip. Okay, there's a diagram on the internet that can help you. Seek, someone answer, seek. C is for Citrobacter, E is for E. coli, Enterobacter, K is for Klebsiella, S is for Serratia. These are the lactose fermenters. Okay, you can say KC, seek, yes. So there, these are the answers. Okay. Now, pleomorphic basically means they have different shapes, but on classification, let's use these four. Okay. I hope that's clear. Any question? I hope that's clear. Now, this is also a very high-yield topic, the classification, because it enables you to know how to organize your reading. Okay. Now, this one, I saw many people couldn't tell. So this is the topic of sterilization. So I know there are some topics you guys don't like reading. I also didn't like reading this topic, but at least know something. Know a few things. Is Helicobacter spirochete? No. Spirochetes are Treponema, Borrelia, Leptospira. Those are the spirochetes. Okay. So let's know something. Okay. Now this device, what is the name of this device? Autoclave, very good. This is an autoclave. Then mention 4 disinfection methods. So this was also direct. How do we disinfect? So you can use ultraviolet. You can use gases, for example, formaldehyde gas. You can use filtration. Then you can use chemicals, okay, such as alcohol, aldehydes, hydrogen peroxide. Okay. Next, identify 2 examples of bacteria that are normal flora in the following sites. This is also another topic. People neglect normal flora. Then identify sterile sites in health with no normal flora. There's one bacteria you can never go wrong that occurs in almost all these systems. Staphylococcus aureus, yeah. So Staphylococcus aureus skin strep species. So Staphylococcus epidermidis is found on the skin. Strep, Haemophilus. We have upper respiratory tract, staph, Corynebacteria, GI, Enterococcus, strep, Lactobacillus, E. coli, Klebsiella. GI, this is GI, GIT, then this is the genital tract, Lactobacillus, Streptococcus, Agalactiae. Then sterile sites is where you expect no bacteria. Okay, this is a very good MCQ questions there. They ask you, the following are sterile sites except, okay. We have the blood. This makes sense because if you have bacteria in the blood, that is bacteremia. In the CSF, we don't expect bacteria in the CSF. Pleural fluid, pericardial fluid, peritoneal fluid, synovial fluid, at the bone level, at internal organs. You don't expect any bacteria at any of your organs. And then urinary bladder. Okay, so the bladder is sterile, but the urethra is not. Okay. Yes, hope that's clear. Internal body organs here, we're thinking about the liver, the pancreas, the spleen, okay, the kidneys, those are sterile. Okay. Is testis sterile? Yes, the testis is sterile. Unless proven otherwise. Okay. Identify the above characteristic appearance on GMSA. So for this picture, I had to zoom, but can you see this thing? What shape is this? This is the picture I was targeting. Sorry, because of the zooming, it's not that clear. But what do you think? What do you think is this? Safety pin appearance. Very good. That was what I was targeting. It looks like a safety pin. Okay. What about ascending infections in UTI? I don't understand that question, but if you mean when the bacteria ascend to the bladder, it now becomes infectious. At the urethra level, it's okay, but when it starts ascending, it's now starts to become infectious. I'm seeing you're asking for screenshots. I'll send you the PDF. Yeah. 2 bacteria with the above appearance. So this is safety pin appearance, very specific to which bacteria. Then what is the gram reaction and shape of the organism shown? Yersinia, very good. Okay. So safety pin appearance or bipolar appearance, you can see the 2 poles. And then Yersinia pestis, Yersinia enterolytica. And then gram stain, this is gram negative, Coccobacillus. I think we had seen that there. Okay. Now this is another important slide. Okay. Now here, this is directly from your notes. This forms the basis of classification of Staphylococcus and Streptococcus. But now here we're talking about the Streptococcus species. Now we start gram-positive cocci. It can either be staph or strep. Then what test is this that you do so that it's positive in staph but negative in strep? So this is very, very important. Answers? I'm seeing people saying coagulase, some are saying catalase. It's actually catalase. Okay, coagulase, please don't confuse. You see, for the pathway of staph, we go like this. Staph, then we do coagulase. So these bacteria, if you can see this appearance, this is in clusters. This is in, what shape is this? Pairs, or it's a chain actually, not pairs. It's a chain, and this is in clusters. Now for staph, in order to differentiate between Staphylococcus aureus and Saprophyticus and Epidermidis, we now do the coagulase test. Staph aureus is positive always. That's how I remembered. So catalase positive, coagulase positive. Okay. Now for this other side, you do the catalase test. It gives you this chain appearance of, so choice B is Streptococcus species. Then we grow on blood sheep agar. It gives you 3 types of hemolysis. We have beta hemolysis, we have alpha hemolysis, and then the third one is the no hemolysis. So it's not here, but that's something you should know. Okay. Please, if people have questions on the chat, let's answer each other so that we move faster. Unless like there's a burning question where I can answer at the end. Okay. But let's answer each other. Let's share knowledge. Okay. Now here, we start with the beta hemolysis. Now for beta hemolysis, there's a mnemonic I usually have for the beta hemolysis. Let me write it down. So I have so take note of this. We have B RAS, okay, and then the other one is usually overpass like that. Then BB RAS, what does that mean? So BB RAS means bacitracin. So you can see here in beta hemolysis, we use bacitracin. Now this bacitracin, the mnemonic goes like this. Group B resistant, group A sensitive. Have you noted that down? Group B resistant, group A sensitive. That's BB-RAS. Then this other one, overpass, which is for optokin, that's for alpha hemolysis. You say optokin viridans resistant, and then pneumoniae sensitive. So take note of that. So you say optokin viridans resistance, pneumoniae sensitive. So from that mnemonic, BB-RAS, beta hemolytic, bacitracin. So here it says sensitive. So sensitive will be group A sensitive. Okay. Then resistance is group B resistance. Now here is alpha hemolytic. Alpha hemolytic, we use optokin. So the answer is optokin. Then here we have other alpha hemolytic, which are the resistant, which is viridans. And then here we have strep pneumoniae, which is F. Okay. Then you can add the other one where it is the no hemolysis. The no hemolysis is where we have Enterococcus. You remember them? Enterococcus faecialis, Enterococcus faecium. Okay. Yeah. So here they are. Catalase, Streptococcus species, Streptococcus pyogenes. You can write Streptococcus pyogenes. Okay. This is BB-RAS, group B, group A sensitive. So you can write pyogenes or group A, and then here you can write group B or Agalactiae. And then here is optokin. Okay. Then F is strep pneumoniae. I hope that's clear. This is very, very important. Okay. But Enterococcus group can be alpha or gamma. Is Enterococcus alpha or gamma, or is it no hemolysis? Gamma, no hemolysis, yes. Aman, you want to unmute?
Yes, Nathan.
Yes.
I was just saying one thing that Medicosis Professionalis said that Staphylococcus and Streptococcus we can use coagulase test. Staphylococcus is coagulase positive, Streptococcus is coagulase negative. So now I'm actually wondering because that's like a known guy. We said that we can't use coagulase.
What does the literature say?
Which literature? Which book?
I mean, your notes, your lecturers, any microbiology group book. You said coagulase.
Yeah, coagulase.
Okay.
You can even refer from his video. Now I'm actually wondering.
Oh yeah, so that's a good point, because you see, when we want to differentiate these two, the reason we use catalase is because all Staphylococcus species are catalase positive. Not all Staphylococcus species are coagulase positive. Okay, that's why we use catalase rather than coagulase. Is that clear? Does it make sense? Yes, you okay now? But let me even check that real quick. Streptococcus coagulase test. So Streptococcus is coagulase negative. Yeah, it's coagulase negative, but you can't use it to differentiate both, you know, the two. Okay. Sorry, I was unmuted. Can you hear me now? Give 2 examples of antibiotics that work in the following ways. So I chose, it was very random. I would have asked anything, cell wall synthesis inhibitors. So please go through that. Mode of action of antibiotics, mode of action of antifungals. Those, if you don't get antifungals, you'll be asked antibiotics. If you don't get antibiotics, you'll be asked antifungals. So we start with protein synthesis at the 30S, 50S, and nucleic acid synthesis. So yes. So yeah, I'm seeing answers, very good. Higher. 30S, it usually goes these 2 are pairs, and it can help you in microbe and in pharmaco. Aminoglycosides goes with tetracyclines. Remember that, 30S. Then 50S is where we have a lot. We have macrolides. Macrolides are the ones that end in mycin, erythromycin, clarithromycin. Okay. Don't confuse with clindamycin. Clindamycin is a licosamide. Then we have chloramphenicol. We have linezolid. And then inhibit nucleic acid synthesis. These are the ones that interfere with DNA and RNA synthesis, which are the fluoroquinolones, like ciprofloxacin. Now, I want to ask you guys from here. We have, let's use staph, because I want to ask that. Now, staphylococcus, what is the treatment of staphylococcus? If you're just asked generally, what do you use to treat staphylococcus? Staphylococcus aureus, that is. If you're given a slide and the answer is staphylococcus and they asked you what is the drug of choice, what will you say? Penicillin, now that is where I've caught you, penicillin. So you see, that's, you fell into the trap. Dependent on resistance of species, yes. But now you see staphylococcus, most staphylococcus are resistant to penicillin. Okay, even like in clinical practice, you'll never see, after culture, you find staphylococcus, you never see them use penicillin. Okay, but now since this is microbiology, we have MRSA and VRSA. So we have methicillin-resistant Staphylococcus aureus, where we use vancomycin. What about if we have vancomycin-resistant Staphylococcus aureus? What drug do we use? Linezolid, very good. So regardless of, no, you cannot use methicillin again. It is already methicillin-resistant. Okay, so regardless, so if you're told first-line treatment of Staphylococcus, think of vancomycin. Then if it's, because we are already assuming it's methicillin resistant because there are high rates of resistant. Okay. Now, another thing they can ask you, what is the mechanism behind the resistance? What is happening? Why is MRSA resistant? That's also a potential question. Yes, what will you say? Why is there resistance in MRSA? Antibiotic abuse? No. MEK gene present, very good. So the MEK gene present, it's actually MEK A gene is due, the resistance is due to altered penicillin binding proteins. Okay, these penicillin binding proteins. Yes, very good. Okay. What about cloxacillin? Yeah, anything you see ends with ilin, it is in the penicillin aspect of drugs. Those are all penicillins. Pardon the reason. So the reason is we are having resistance due to altered protein binding. penicillin binding proteins. And this is conferred by the MEK-A gene. Okay. So I hope that's clear. Take note of that. I don't want to get into details because we can discuss Staphylococcus a lot and it has a lot of things, but I'm sure we're going to get a question similar to that. So we discuss that. Okay.